So really the goal today is a bit two different things. One is to show you a really nice example of how a QSP model could really help to inform drug development. And the other one is to show you how to do that with the MonolixSuite, so a bit as a MonolixSuite demo. So I will first give you a bit some background information about the molecule we are looking at, which is a FAAH inhibitor. What's the question that we would like to address? And then I will really show you a bit step by step how to proceed. So first, how to implement the model using the ML language that we use in the MonolixSuite, how to format data in case you would like to estimate parameters based on some longitudinal data. We're going to also have a step of exploring the model using our Simulix application that is meant for simulations. And then finally, how to estimate parameters with Monolix. So that's really the model implementation part. And then we have a model prediction part where we're going to use the model to predict the PD effect at different dose levels. So I would like to stress that like the story behind this case study has not been developed by us. I really borrowed it from a publication. So what we are going to look at is actually an enzyme that is called Fatty Amide Hydrolase, so let's call it FAAH, and it degrades fatty amides such as anandamide that we're going to call AEA. And actually we are interested in this fatty amides because AEA binds to the cannabinoid receptor CB1 in the brain, and actually this binding decreases the perception of pain. So what we're going to look at is a drug that is a reversible inhibitor of this FAAH enzyme that is developed for osteoarthritic pain. And the idea is that if the drug inhibits FAAH, we're going to have less degradation of AEA, so more elevated AEA levels that will bind more to the CB1 receptor and that should decrease the pain sensation. So that's the idea of this drug. So that we already have some preclinical data, and actually the results in the preclinical species are a bit contrasted depending on the stimulus intensity and the disease phenotype and the species. We can have a bit contradictory results. This drug, nevertheless, was pushed to phase 1, and actually what we can see from the phase 1 clinical data is that if we look at the FAAH inhibition in plasma, it is really highly inhibited already with doses as low as 0.3 milligram, both with single and multiple doses. And if we look at the AEA in plasma, we see that that's like base level. And then when we give the drug, we see that the AEA levels are really increased, and then they come back when the drug disappears. But we really have an effect on AEA and on FAAH inhibition. So that's what we were looking for. So now we are really in the status between phase 1 and phase 2. And the question that we are asking ourselves is, first, do we understand the biology well enough to proceed to phase 2? And second, which dose do we need to elicit an effect? And for the moment, we have data on AEA elevation, but that's not really an physiological effect. So we would like to try to simulate a bit in terms of effect what we could get for different doses. And to do so, the idea is to develop a QSP model to capture the known biology, and then to use this model to simulate the effect of different doses. We will not be able to really go up to the simulation of the pain cessation, but what we will look at is the CB1 occupancy as a surrogate of the response. So if we want to develop a QSP model, let's a bit look at what we know about the system. So what we know is what I have already shown to you previously. What we also know is that actually AEA is produced in several steps. So there is a precursor that we can call NAPE that itself is produced from phosphatidyl ethanolamide. So that here I call PE. Actually, well, I'm going to put my mouse a bit more visible. So maybe we could take into account also these steps that enable the production of AEA. And another important aspect is that actually FAAH also act on other fatty amides. There are like 4 different ones that here have lumped just with the letter X. And actually, there is a competition for the action of FAAH. So it could be interesting in the model to take also these other fatty amides into account. So that's a bit about the different reactions that are really key in the process we are looking at. And then we can also take into account a bit the physiology. So what we are really interested at is what happens in the brain with the binding to the CB1 receptor that is really in the brain. But actually the XEAs, or all 5 of them, are lipophilic, so they can cross the membranes, and we can also find them in the blood-brain barrier, in plasma, and in the rest of the body. So maybe we can take these 4 different compartments into account. And what we also need is to capture the distribution of the drug in the different compartments that we can do with a smaller PK model. So that's a bit the framework of the model that we would like to develop. Now let's assume that for all these reactions, I have written my equations on a piece of paper. What I need to do next is to actually write them into a language that Monolix can understand. And that's why I'm going to explain you a bit how the MLXtrain language works. So MLXtrain is really a language that is understood by all the applications of the MonolixSuite. We have a quite big model with many reaction rates, ODEs, parameters. I will just show you a small extract of it to give you the main key aspects. So that's a small extract, and you see I have a lot of ... that just indicate that I have like really skipped some parts out. So in this model definition, I first have this keyword longitudinal that I always need to have at the beginning of the file. Then I have a line input. That's where I'm going to put all the parameters that I would like to estimate by fitting the model to some data. Then I have this PK block. That's a reserved syntax. And what I can put in the PK block is macros. So macros are like similar to functions, actually. And for QSP model, there is one macro that is interesting, and it's called depot. What the depot macro does is that it's going to read the doses that are defined in the dataset and apply them to the ODE variable that is defined in the target argument. So here, basically, all doses defined in the dataset will be applied to the ODE variable, a drug that represents the amount of drug in plasma, for instance. Here I have a very simple use of depot, but actually the depot macro has additional arguments like Ka, Tlag, P, that allows you to define first order absorption, lag times, bioavailability, etc. Then I have a block equation. In the block equation, that's where I'm going to put ODEs. And actually, we have 2 different ODE solvers. By default, it's a non-stiff solver that is used. Usually, QSP models that are like big models with different timescales for different variables are stiff. So if you are working with a QSP model, it's important to add this line, ODE type equals stiff, such that the stiff ODE solver will be used. Then here I have some lines where I just defined a value for some parameters that I will just consider fixed in the model. So I just give the name of the parameter equal its value. And here you can also do parameter transformations, etc. Then I have a part where I define actually the initial status of my ODE system. So I need to define the initial time at which I'm going to start to integrate the ODE system. In this, most of the time, the initial time will be 0. In this example, actually, I have decided to put a negative value. And the idea is to say that I don't know precisely what is the steady state value of all my ODE variables, because that's really a complex model. So what I'm going to do is just to give for each variable an approximate value. And then I will start to integrate the ODE system, so all variables will move and stabilize to their steady state value. And then at time 0, I'm going to give the dose, and then I'm going to apply my perturbation to the system. So that's just me deciding that the dose will be at time 0, and I start to integrate before to have time to reach steady state. Just below here, you see the definition of the initial value for 2 different variables. So to define an initial value, we use the syntax _0, and what we have before is the name of the variable. So here my variable is called A_P and O_P. In this part, I have the definition of the time derivatives of my ODE variables. And here the syntax is DDT_, and then the variable name. And then you just put the equation as you would do in any other software. If you need to use power, it would be this small symbol here. And then we have many functions like exponential, log, max, absolute value, etc. And then at the very bottom of your model, you will have a section output. And in the output line, that's where you define all the ODE variables or all the variables from your model that you want to map to the observations you have in your data set. That's a bit an overview of the key things we need to know to implement this specific example. Just to give you a bit some additional options. So there are a couple of additional keywords that can be quite convenient. So T is our keyword for time, in case you want to define, for example, a time varying clearance. And then we have some keywords to recover dose information. So AMT dose is the amount of the last dose. And that can be convenient if you want to define the dose-dependent bioavailability, for instance. And it's also possible to use if-else statements. So it really gives you quite some flexibility to implement even like pretty big QSP models. So that's what was a bit an overview on the slides, like really with the extract of the model. Now let me show you like the full file, how it looks like. So I have my longitudinal keyword, and then in the input for the moment, I really have put all parameters. It's really a lot. Probably I will not be able to estimate all of those. We're going to see a bit later what we can fix. Then I have my block PK, my block equation. And then here I have a lot of lines that define all the reaction velocities. I have many different reactions, so that's a lot. And then here I have the initial value for all the ODE variables. And then here I have the time derivatives for the ODE variables. Here I have opened so the model is just a TXT file. I have opened it using the MLX editor because the MLX editor includes a syntax checker. So for example, here, once I'm done writing the model, I can click on check syntax so that I get a check of is everything properly understood. If everything is properly understood, I have this green message, everything okay. And in case I'm doing a typo, for example, if here I just forget the last letter, when I click check syntax, I will have a nice error message saying at line 58, which is highlighted, I have this variable that is undefined, so not known because it's not defined previously. So really, this MLX editor allows you to write your model and also check with nice error messages that everything is properly defined and understood. All right, so that's about our big model. Let me go back to the slides. So we have seen in our model that we have many different parameters. And so among them, I have PK parameters, organ volumes, some partition coefficients, some distribution of enzyme or precursors among different organs, reaction velocities, inhibition constants. We will not be able to estimate all of them just with the phase 1 data we have. So what we can do is to look a bit for parameter values in the literature. Maybe we can also find some parameter values from some in vitro experiments, for example, some binding assays. And then for the ones that remain, we're going to try to estimate them by fitting the model to the phase 1 longitudinal data we have collected. Let's first have a look at what we can find in the literature. And actually here I have a bit an overview of everything that we could find in the literature. So that's especially Vmax and Km for the enzymes, also the distribution of enzyme across different organs. As you can see, quite often, actually, we have data from rat or mouse, but not human. But that's what we can take at least as a first case. So basically, for all these parameters, instead of having them in the input line of the model, I'm going to have them defined directly in the body of the model with the name of the parameter and its value. Then I also have actually some PK data from the phase 1 study. So what we could do is to handle the PK data a bit separately, fit a PK model to the PK data. That's what I have done here. And then the estimated parameters, put them also in the model file. So these are fixed. And then when I'm done with all this, actually I have a couple of parameters that remains in the input section here. And those I have no information so far in the literature. So what I'm going to try to do is to estimate them based on my phase 1 data. So talking about the data that we have, so what we have collected is actually the XEA concentration. So for 4 out of the 5 that we know exist over time, after a single dose of 1 milligram or a single dose of 10 milligram. So that's the concentration over time. And then we also have the FAAH activity relative to its baseline, also for the 2 different doses that were tested. So we can use this data to try to estimate the parameters we have left. Before we can do that, what we need to do is actually to put this data into a file format that can be read by the MonolixSuite. So let me tell you a bit about the dataset format. So when we work with the MonolixSuite, we need to work with what we call a long format, so where each row is 1 time point per subject. And then we have all the subjects on top of each other in a long format. The file format itself can be a TXT, CSV, Excel, or SAS. We need to have 1 header line, and then all different types of observations need to be in 1 single column. So if we look a bit more in detail into our example, so we have dose information and observation information. So we would usually separate dose lines and observation lines. Here, for example, I have the data for individual 1 with the different time points, and have a dose line where I indicate the amount. Actually, that's a bit a separate example because in our case, we have 1 and 10 milligram. And then I have a column with measurements for each of the different time points. To distinguish the dose and the observation lines, I can either use a dot in the cell that is not meaningful on that line, or if I prefer to have numeric values everywhere, then I would need an additional column called EVID that has value 1 to indicate those lines and value 0 to indicate measurement lines. Then what we can also think of is actually how to distinguish different profiles. And in Monolix, it's going to really be the ID column that will allow you to group together measurements to indicate that they belong to the same profile. And then this profile can be individual data, or it can be average. This does not really matter. Just remember, 1 ID is 1 profile. So in our example, we don't have individual data, we just have average data. And actually we have the average data for those 1 and those 10 milligram. So I have created 2 different individuals in the data set, with individual 1 corresponding to the dose 1 milligram and individual 2 corresponding to the dose 10 milligram. And then when I look at the data in Monolix, I will see 1 line for each of the individuals of the data set. So in our case, for each dose. And then in our case, we also have different type of measurements. We have measurements of the XEA, and we have several XEA, and also for FAAH. So what I need to do is to put all measurements in the same column, and then have a separate column to indicate on each line what I have measured. In this DVID column, I can have either integers or strings, and read to indicate, okay, that's a measurement for LEA, and then I can have at the same time another value that represents a measurement for FAAH, for instance. And then this column, we're going to indicate it as being observation identifier in Monolix. All right, so now that we have seen how to write the model and how to define, like put together the data set, let me show you how to put all this information in Monolix. That's what we're going to do next. And to do so, I'm going to open the Monolix application. When I open the Monolix application, the first thing I need to do is to give in the data file. So I'm going to browse and select on my machine the CSV file that corresponds to what I have shown to you on the slides. Each of the columns need to be tagged so that Monolix knows what it is. Some columns are tagged automatically. So here I have column ID, time, DV with all the observations. DV ID is tagged as observation ID that distinguish the different things I have measured. Dose is not tagged automatically. Actually, that's a column that indicates like the two different groups, dose 1 and dose 10 milligram. I can tag this as categorical covariate. It's going to allow me to like color all my plots or split them by dose. And I have the amount on the amount lines and my EVID column to distinguish dose and measurement lines. Once I click on accept, the data set is loaded, and I can right away have a look at it in the plot tab. In the plot tab, I'm going to have one separate plot for each of the species indicated in the observation ID column. So here I have the two different profiles for AEA. In the stratify tab, here I have only dose, but if you have like several continuous or categorical covariates, you can really split color or filter for all these covariates. In my case, for example, I could split by dose and display the legend to better identify what is dose 1 and what is dose 10 milligram. So that's about the data. If I go back to the structural model tab, that would be the second step, is to load the model I would like to use. So I have saved the model as a txt file before, so I can browse for it and select this model 1. If I go back to the structural model, so I see here that's really exactly the same as what I have shown to you before. And actually, what I have at the very bottom of this file is the different variables of the model that I want to map to the observations I have in the data set. And that's a panel where I'm going to indicate this mapping. So in this column, I have everything that has been found in the observation ID column of the data set. And here, everything that is listed as model output. And then I can drag and drop to indicate what is what. So here I'm just basing myself on the different names that I have. So OEA, actually I call it O_P, P for plasma in the model. So I'm just doing all the lines and clicking accept to say what corresponds to what in terms of model and data. Then I have a tab where I can indicate initial estimates. We're going to go back to this later. And a tab where I can define a bit more the statistical part of the model. So here I can see all the different observation types, and I can choose a residual error model for them. And below, I have all the parameters I would like to estimate. In my case, I don't have any individual data. So actually, I just would like to estimate one Vmax NAT value. So what I'm going to do is to remove the random effects. Random effects are convenient if you want to estimate a different value for each individual to include inter-individual variability. In our case, as we work with average data, I will just remove all of those. And then before I start the parameter estimation, I need to choose some initial values for my parameters. By default, they are all just 1. And here the question is a bit, okay, I have a number of parameters. I'm not so sure if I will be able to identify all of them. For the moment, I don't really know what's the impact of each of these parameters on the observations I have in the data set. And actually, to learn a bit more about this, we have this tab, Check Initial Estimates, where we can play with the values here at the bottom. And then at the top, we see in blue the data, and in red, the model prediction using these parameters. And then I have one tab for each of the different measurements I have in the data set. And here, for example, I could play a bit with the values and see how that impacts the curve directly on the plot. That's pretty nice, but it can be a bit limiting if you would like to actually view all different observation types at the same time. So to have a bit more flexibility to look at the impact of the parameters, what I have done is that I have created a Simulix project. So Simulix is our application really to perform simulations of any kind. It's very flexible. And what I have done is that I have really reused the same model file, defined some initial parameter values I want to start with, defined the base treatment, so 1 milligram single dose, defined among all variables, defined the model, which one I would like to plot. And then in the exploration tab, I can really see the simulation for all my variables I'm interested in. So that's really the ones that have been measured in the data set. And I can play with the parameter values really in real time to see the impact on the model prediction. To better see the impact, I'm going to click here, mark current as reference, so that I freeze what I have on the screen. And then I can check, okay, if I increase Vmax NAT, what's going on? I see that actually the concentration for each of the fatty amides is decreased, like the Cmax is decreased. We have no impact on FAAH. And all of the fatty amides are decreased in the same way. So this parameter really impacts all variables in the same way. And actually, if I have another parameter where I have one parameter per each of these fatty amide, that when I change it, it's going to change only the corresponding fatty amides. If I explain this to you a bit more on the slides. So Vmax is really common to all equations affecting each of the fatty amides. And then for each fatty amide, we have a separate scaling factor that allows you to have different maximal velocities for each of the different fatty amides. And then we have the degradation of FAAH, that is a separate parameter we would like to estimate. We have the inhibition of FAAH, so that's how strongly the drug inhibits FAAH. FAH underscore T represents the steady state value of AAH when there is no inhibition by the drug. And then these two factors, let's call them, allows to have a different inhibition by FAH for each of the fatty amides. So in the parameters we would like to estimate, some are like parameters that are common to all fatty amide, and some allows you to have specific values working more as a factor. So here we could really spend more time playing with the parameter values. When we play with them, we can really see that except the factors for which we need to fix one, because they are really relative to each other, so one needs to be fixed to be the reference, all others have actually a different impact on what we can observe. So they all have a good chance to be identifiable. All right, so actually, if all parameters seem to be identifiable, what we can do is to proceed with our Monolix run and try to estimate all these parameters. This takes around like 10, 15 minutes to run on a laptop. So I'm not going to do it in front of you. What I'm going to do is to load a Monolix run where all the results are already available. So in the Monolix run, it's exactly what I had before. So I have the data set, the model. I have put some initial values that I have chosen thanks to this check initial estimates tab. Some of the parameters appear in orange because they are fixed. So that is like factors that we use as a reference. And then I have clicked run. I have waited a couple of minutes, and then I get the results as a table. So here I can see the estimated values and the relative standard error. We can see that the parameters related to the degradation and inhibition of FAAH are pretty uncertain with high relative standard errors. And then I have the plots. To see the plots, I need to click on the plot button to see them again. And in the plot, then I have all typical diagnostic plots. So I can see the fit of my model to the data in blue, have a plot observations versus predictions, etc. I'm going to have one plot for each separate type of measurement so that in order to see a bit everything at once, let's go back to the slides. And actually, that's the fits we would get for each of the fatty amides with this model. And what we see is that it does not look great. I mean, clearly the model is not really able to capture the shape of the data. So one possibility is that actually our initial values were like pretty bad. And during the estimation process, we didn't manage to find the global minimum. We stopped into a local minimum. To test this hypothesis, what we can do is instead of doing a single run from a single set of initial values, we can run several times starting from different initial values. And actually, this is very convenient to do with Monolix. I can just go here to the tab Assessment, and here I can, in one click, run as many runs as I want, with that each will start from different initial values. And if I show you the results on the slides, actually, we would have an output like this, where here I have done 10 different runs that you see on the x-axis. And then for each run, we see the estimated value as a dot and the standard error as a bars around. And what we see is that among the 10 runs, we have kind of 3 outliers for which we estimate a different set of parameters. But for all others, actually, we find the same estimate that lead to this fit that is really not great. So maybe there is something that is missing in the model. Let's try to interpret a bit more what's going on. If I look at the data, so that's my initial value for, let's take one of the fatty amides. Does not really matter, they all have more or less the same shape. So the initial value, sorry. If I look at a small extract of the model, the fatty amides are produced and degraded. So if I don't have any drug in the system, they have a production rate, a degradation rate, and then this gives me the steady state value. And then when the drug inhibits FAAH that degrades the fatty amides, I'm going to actually change the degradation rate because the enzyme is inhibited. And this is going to give me a new steady state value in the presence of the drug. That corresponds to the same production, but a degradation that is reduced by a fraction that is as much as the inhibition of FAAH. And actually, we know by how much FAAH is inhibited. We know that that's around like 97%. And if we apply this in the equation, actually the new steady state value for the fatty amides when the drug is present in the system is actually 33 times bigger than the initial value. And that's not at all what we see in the plot. In the plot, the legend here is a bit small, but that's value 10 and that's 50. So if it's 33 times bigger, it would be more over there. And actually what the model is doing is that if we would let the drug present in the system for a long time, actually we would start from this initial value, go up to there, stabilize, and then decrease again when the drug disappears. Because the model that does not correspond at all what we have in the data. What the model going to do as when it estimates the parameters is that it's going to let this concentration increase very slowly, so that actually it does not have time to reach its new steady state. And then because the drug is cleared from the body, so the drug disappears, then it's going to go back to the base level. So basically the slow change of the fatty amides is a bit a solution that the parameter estimation algorithm has found, such that the concentration is not too far away from the observation, but in terms of shape, we see that that's not really what we want. So there is really something that we miss. And if we think, okay, actually, from this initial value, we go to a value that is around 5 times faster, higher, but not more, it means that actually, even when FAAH is inhibited, I still degrade my fatty amides quite a lot. I degrade it a bit less, so that it increases 5-fold, but not as much as 97%. That would make an increase more around 30-fold. So in order to take this into account, it really looks like we have actually another degradation mechanism for our fatty amides, so a second type of elimination that is not related to FAAH, such that even when FAAH is almost completely inhibited, we still have this second degradation that takes place and that allows an increase in the fatty amides, but not too much. So it really looks like that's something that we missed at the beginning, but that is quite important to understand the range of increase in the fatty amides that we see. So what we can do is actually to look in the literature for what could mediate this second degradation. And so we are looking for a second enzyme that would degrade our fatty amides. And when we look into the literature and also looking a bit at the binding affinities, so that we are in a range that makes sense with what we see experimentally, it looks like that the best candidate is an enzyme called NAAA. And this one is able to degrade the different fatty amides we have in a concentration range that is consistent with what we observe. So what we can do is actually to develop a second model where we introduce this second enzyme to degrade the fatty amides. I'm not going to show you all the details. That's just a small change in the equations. What I'm going to do is to directly show you how this second model with just one additional enzyme is able to fit the data. And here you see the fits for all the fatty amides, and they look much better than before. It's not perfect, but we are able to see the increase, then the plateau, and then going back to the base level, especially for the higher doses. So that's a parameter estimate. What we also see with this second model is that actually we have a much lower RSC value, so a much better confidence in the estimated value. That actually is because our model captures better the data. All right, so now let's come back to the questions that we had at the beginning. So the first question was about understanding the biology, and we have progressed into this. We really have tried to put all our knowledge around the molecule of interest into a model. We have seen that actually we missed one of the key enzymes. We have added it to the model, and now it looks already much better. Now, the second question was, which dose do we need to elicit an effect? And you remember that actually we are interested in the effect in the brain, where the binding of the fatty amides to the CB1 receptor will decrease the sensation of pain. So we cannot, it would be very hard to really model directly the effect on the pain levels. But what we can do is to look at the CB1 occupancy as a surrogate of the response. So that would be how we could define CB1 occupancy, depending on the concentration of the AA fatty amides in the brain and the KD for CB1. And actually, I'm going to show you how to do these predictions. And to do that, I'm going to open the second run that I have done with an AA. I'm opening that in Monolix. So here, that's all the results I have shown to you directly on the slides. And if I would like to perform simulation based on this model with these estimated parameters, what I can do is to export this project to Simulix, which is our application meant for simulation. So I'm exporting, and that's going to open a Simulix window. In this Simulix window, I'm really going to have all the information that comes from my Monolix run. So I have, for example, all the models that is defined here. I have the parameters that I have estimated, etc. And what I can define by myself is what is new. Actually, what is new is that I would like to simulate the CB1 occupancy, which is not part of what I was usually looking at. If I look in the model, actually I have a variable called CB1_B for brain. That is the equation I have to shown to you on the slide. So that's my CB1 occupancy. So I would like to output this. So to do so, I go to the output, create a new output. I would like from everything I have in the model to output CB1. So that's CB1 occupancy. And then I can choose a time interval over which I want to simulate. So let's say up to 300 hours. Click OK. And then I can also define maybe different dose levels I would like to simulate. So for this, I can go to the treatments. And here again, I can, for example, check for a dose of 1 milligram. So that would be 1 milligram at time 0. Then I will do another one that would be 10 milligrams, so single dose of 10 at time 0. And maybe also 100. Once I have prepared everything I want to use for the simulation, I can go to the Exploration tab. And here I'm going to select the different doses I have defined, each in a different exploration group so that I can see them as separate lines on the plot. And then for the outputs, I'm going to remove everything I have here and select CB1 occupancy. And when I click on Apply, I'm going to perform a simulation, outputting the CB1 occupancy as a percentage for the 3 different doses. And what I see here is actually when I increase the dose, it looks like I can have a CB1 occupancy that lasts for longer. But actually, in terms of maximal level of CB1, even for pretty high doses, I'm stuck at 10%. That's really the maximum I managed to get, even for high doses. And that's pretty disappointing because actually that's maybe not enough to really decrease the pain sensation in osteoporosis arthritis. So that here on this plot, I have like simulated with some more dose levels, but really the conclusion is that even for high doses, maximal CB1 occupancy is 10%. Then the second question is actually, how confident can we be in this prediction? We have a lot of parameters that we have just fixed, for which we are quite uncertain about the value. So maybe we would like to check a bit how the CB1 occupancy would change if all the parameters I have in the model are actually a bit different compared to the value I have assigned to them. And to do so, I can do a sensitivity analysis. And what I would like to do is for each of the parameters in turn, change a bit its value and check the impact on CB1 occupancy. I could do that in the graphical user interface, but as I have a lot of parameters in the model, if I also consider the ones that I have fixed, it would be a bit tedious. So instead, I can use the Lixoft connectors. That's the R package that actually corresponds to an API to the MonolixSuite. So all the actions you can do in the graphical user interface, you can also do them via R functions. And these are functions that really use the same C++ code in the background as the graphical user interface. So you are really sure that everything is consistent. So just a small example about how the code in R would look like. So you could really define some elements as I have done in the graphical user interface. So with the treatment, the output. And then for each parameter in turn, we will increase the value by 20% and check how much it changes the CB1 prediction. And then we can, for example, plot this in a graphical way. And actually, among all parameters that we have tested, most of them have no impact or like really a minor impact on the CB1 occupancy. And here I have plotted only those for which we do see an impact on CB1 occupancy. And if we go a bit more in detail into these parameters, Vmax_nat, that's a parameter that we have estimated on the data. And actually, we have estimated it with quite good confidence. So probably this one is quite reliable. A_nat_A, actually, this is one of the parameters we have fixed to 1 because it's redundant with this one. So this we can kind of forget about it. Then the binding affinity of CB1 of the fatty amides to CB1 has also quite some impact. This one is one of the rare parameters that we obtained in human from a real meta-analysis. So this one is, we are also quite confident in it. And KCLA is also estimated from the data. And the other ones are minor. So we remain with those three. And if we focus with B_nat brain and BNNA brain, actually these two correspond to the distribution of the enzyme among the different organs and in particular into the brain. And the value that was used is actually a value that come from measurements in rat, which are themselves quite uncertain. So that's typically parameters for which we have a high uncertainty that then impact our CB1 prediction. So overall, we predict that CB1 occupancy would be quite low, but this prediction is quite uncertain because it depends on parameters that are themselves quite uncertain. So to conclude, actually, we have looked at a bit two different type of questions. So the first one was really more about the mechanism. And what we have found is that our fatty amides are not only degraded by the enzyme we are targeting, FAAH, but also by a second enzyme. And actually that the second enzyme a bit limits the impact of the FAAH inhibitor, because even if we inhibit FAAH, there is still something that degrades AEA, and therefore we cannot increase the levels of AEA as much as we would like. So maybe knowing this, a good option would be to look for therapies that would combine something that targets FAAH and also NAA at the same time to have really a higher increase of AEA. So that is really something that is a nice insight given by the model. And then the other thing we were looking at was to predict CB1 occupancy. And actually here we have seen that, okay, we can do a prediction, but this prediction is quite uncertain given all the uncertainty we have in our parameter values. So on this, the model actually at the end cannot say much. So that's like more the conclusion for this case study. And then I also would like to a bit highlight the advantages of doing a QSP model with the MonolixSuite. And actually, it really offers you an entire framework that gives you many different tools to write your model in an efficient way, estimate parameters, but also give you tools for checking the identifiability of your parameters, either doing some manual sensitivity assessments, as we have seen in Simulix, but we also provide more advanced functions like profile likelihood or things like this in separate R packages that work with the MonolixSuite in the background. So really a nice framework for QSP models.